So for exam 2 review, let's go through some of the logistics. So your lecture exam, same thing as last time. Same range in which they're available. So they're available from Sunday to Monday at 11 p.m. Central Standard Time. The lecture has 50 questions. The first set of questions that you'll see, well, technically 51 because there's that one question at the beginning. But essentially there's 50 questions on the content. And of those 50, 40 are multiple choice. And 10 at the end are fill in the blanks. Okay. And they will be divided evenly. So I have about 1, 2, 3. Now muscles technically has 2 chapters and nervous has 2 chapters. So I have 1, 2, 3, 4, 5, 6. So I'll divide it up by 6. So you can see about 8 questions on joints, 8 questions on skeletal, about 6 to 8 questions each. So that's going to be about 10 to 12. And then 10 to 12 on the nervous. Okay. And so the muscles and nervous will be more detailed because they do involve multiple chapters than the skeletal and the joints. Any questions on the lecture exam? Okay. Then the lab exam has 30 questions and they are all fill in the blank. Or, typing. Okay. Identification. Remember, no abbreviations. If there's a pair of something, make sure you indicate whether it's right or left. No abbreviations. Read the questions carefully because it could ask you for something general or it could say specifically, what bone is this? So rather than just saying the pelvic bones, you would say, for example, the pubic bone. Okay? So read question carefully. All right. Any questions on the lecture or the lab exam? Okay. For both of these, you want to make sure that you do you have to use respondus lockdown. And just as a reminder, make sure that you check that everything is working by opening up the practice quiz, because the practice quiz is something you can take anytime. And the reason why I did that is so that you can test the computer that you're on, whether it's compatible and it's working fine or not, and your internet is stable enough to support it, okay? So make sure you're using the Respondus Lockdown browser plus webcam. And it's really important that you do the environmental scan. And I showed you a video. I usually attach a video of how to, what is expected in the environmental scan. There are a few cases with the first exam where they didn't do appropriate environmental scan. So I had to, dig further and ask them about. So just make sure that you do a proper environmental scan because I do check these. So the environmental scan is where I want to see what's at your table, what's at your surrounding area, and make sure it's clear. There's no cell phone, there's no notes. You can have a blank piece of paper, and in your environmental scan, that's where you'll show me the front and back of your blank piece of paper. And then at the end, you'll also show me that piece of paper one more time, okay? Any questions on the logistics of the respondus lockdown, the webcam, and the environmental scan? Okay, So that's pretty much the overview for the exam 2, the lecture and lab exams, how you need to have respondents lockdown, webcam, and then do a proper environmental scan. All right, so if there's no questions, I'll move forward. For the skeletal system, certain things that you'll want to be responsible for knowing will be the types of bones, such as what's a short bone, what's a flat bone, irregular, and long. Now, it could be straightforward regurgitation of the definition. So, for example, what type of bone it has its height equal to its width? What would you say? What type of bone has its height equal to its width? What would you say that was? Short bone, exactly. Okay. Or it could be more complex questions. For example, the vertebrae bone would be an example of what type of bone. Perfect, irregular. Very good. So those are the types of questions you'd see based on that topic. Okay. Then make sure you know the functions of bone, which include movement. And it's not that the bones can move by themselves, but they're in association with the muscle. So it's a lever system where it's pulling and resisting. Okay. Then another function of bone is stabilization. Gives you that framework so that your skin can hang on and your organs can be nice and compartmentalized. Okay, so it gives you stability. Okay, protection, certain areas where occur where the bone forms like a cage, like your skull protects your brain, your rib cage protects your upper organs like your heart, your lungs, and your pelvic girdle protects your reproductive organs. So protection is another function. There's also red blood cell production because that does happen in the bone marrow. And then mineral storage. Your bone is as strong as it's able to store the calcium phosphate and collagen. Okay, so mineral storage. Now, of the 4 types of bones, we really do focus quite a bit on long bone. And that's why this is, there's a little mark there. So make sure you know the anatomy of a long bone. Okay, so as I'm drawing the structure, see if you can identify what is what. I can probably do that better. Let me use a highlighter. Okay, so let's start with this right here. We'll do that number 1. What is a number 1? What portion of a long bone is that? Yes, perfect. The epiphysis. So that would be the ends of a long bone. And then this portion What would that be? Perfect, thank you. The diaphysis or the shaft of a long bone. Okay. Then this green structure right here, we'll do that number 3. What would number 3 be? Articular cartilage, excellent. And what's the function of the articular cartilage? Because that's where, I think the term articular meaning two bones like meet each other. So cushion between both. Yep, that's good. So we'll say that the function, it prevents friction. So prevents friction between two bones. Okay, and that's why it's called the articular cartilage, because it's the cartilage at the point where two bones articulate or meet each other. Okay, so you're right, it does cushion, but more specifically, it prevents friction between two long bones. Unfortunately, as we age, the articular cartilage starts to wear down. So what kind of condition is associated with aging and pain between two bones that articulate? Arthritis, right? So that's where it comes along. Okay. Then what would this structure be right here? This one. I'm going to call that 4A, and I'm going to call this 4B. So there's one on the outside and there's one on the inside. So let's do 4A first. What would 4A be? And 4B. Let's start with 4A. What would you call 4A? Because it's wrapping around the bone. So what's the term for going around the bone? Close. So epi means on the surface of the bone. So that's a good guess. But what does it mean to go around something? Peri, perfect. So that's the periosteum. So that's the connective tissue that surrounds the shaft of a long bone, and it allows for attachment specifically to the tendons. Okay, so what would that make B then? 4B. Endosteum, very good. Now, the endosteum is pretty important actually, because that's where you'll find osteogenic cells. We haven't talked about those yet, osteogenic cells. But these are the cells that help to, that are mitotic, so they'll help to produce more bone cells. Okay, and that's where you'll find them. So that kind of gives you a clue as to if you were to break a bone, how would a bone heal? From the inside out or from the outside in? And it would actually be the outside in. So it starts from the endosteum and build and repairs inward. Okay, so that kind of gives you an idea of how bone repairs itself. Okay, all right, moving on to number 5. Let's do this as number 5. What's number 5? Be more specific. It contains the marrow, but what is it? Marrow would produce red blood cells. Yep. But what is it called? What is that structure, number 5? Yeah, yes, close. It is the cavity, but we don't call it the marrow cavity. We call it the what? Medullary cavity. Yep. So the medullary cavity would contain the bone marrow. And it's really interesting because the bone marrow doesn't necessarily contain red blood cells all the time. It contains red blood cells when you're younger, but as you age into like your 50s and 60s, it's going to start to change from red bone marrow to yellow bone marrow, which is fat, lipids. And then from yellow bone marrow, it's going to become gelatinous bone marrow. Okay, so it's not always red bone marrow. So red blood cells are not always made in the bone marrow. Okay, the consistent area where you'll find red bone marrow producing red blood cells actually is at the epiphysis. Okay, so we'll talk about that when we get closer to the types of bone tissue. Okay, so you have the medullary cavity, which contains the bone marrow. Okay, and then how about number 6 right here? What's number 6? What's number 6? It actually can go by 2 different names. Excuse me. Growth plate is what it is. You're right. But it goes by 2 different names. Excuse me. So it's called the epiphyseal plates, or the metaphysis plate. So number 6 goes by either the epiphyseal plate or the metaphysis. And you're right, these are your growth plates. So when you undergo puberty, the growth hormone that starts to be produced targets the targets the metaphysis or the epiphyseal plates and causes it to transition from cartilage to bone. Okay, so these are growth plates that are stimulated at puberty and undergoes the change from cartilage. So it's currently hyaline cartilage to bone tissue. And that's what allows people to grow taller, to elongate. Questions? Okay. All right. So now we're going to move on to the microscopic anatomy. So what we just looked at are things that you can see with your eyes. So we call that the macroscopic anatomy or the gross anatomy. And now we're going to move on to microscopic. So things you can't really see with your eye. We're still looking at a long bone. So now what I'm going to do is I'm going to take a cross section. So like this. We're just going to cut this in half. Zip. Okay. So if I were to cut that in half, this is what you would see. Okay, and I'm trying to stick to the same colors. So what would that make this right here? It's actually not the osteon yet, but that's a great point that I'm trying to make. So if I were to take a cross section of the long bone, it looks like an osteon, but it's actually not an osteon. So what you're seeing in the red is actually a cut of the diaphysis, or if you're looking at the layer, it would be the endosteum, which makes this then what? The medullary cavity. Perfect. So that's going to be number 5. Okay. Now, surrounding all of this, that's to blue, you will find smaller circles. And they're not really circles. If you were to look at it as a 3D, just think of like a whole bunch of pencils, you know, forming a tube. So it's a tube made up of smaller tubes. Now, if I were to hold one pencil, could I easily break that one pencil? Could you easily break one pencil? Yeah, of course, right? But if you were holding a stack of pencils, a pretty thick stack of pencils, could you break that? I couldn't. Okay, so usually you couldn't. So you can see that the structure, like the infrastructure of a long bone, making smaller tubes that form a bigger tube is for strength. And that's why your bone is really strong. Okay, so all of these small little dots that I've drawn in here, I'm going to zoom in on one dot. Okay, so I'm going to zoom in on one dot. Now this is an osteum. So along the diaphysis of the long bone, you will find hundreds, thousands of osteons surrounding that tissue. And then you'd have that hollow space in the middle that we call the medullary cavity. Okay, so that's just to kind of give you an idea of how things are structured. Okay, so the osteon looks very similar, which is why it's so confusing. Let's use yellow, because it also is a circle with a circle on the inside. Okay, but now this picture, the second circle, that is an osteon, which then makes this structure right here. What does that make that yellow circle? It's not the medullary cavity. The medullary cavity is the blue circle, but the yellow circle, so the circle inside an osteon, what is that called? That's going to be the central canal. I'm going to put a little letter A here. I'm going to find the central canal on my list. I'm going to put A there. Okay, so that's the central canal. Okay, now an osteon has these little pockets. Little spaces, little pockets. What are these pockets called? What are these pockets called? Lacuna, very good. Because think of it as lacuna, lake. It's a little lake. Okay. Lacuna literally translates into like a little lake because it looks like a little lake. It's a pocket. And so these things are lacuna. So I'm going to call them B. I'm going to label B under lacunas. Okay. These pockets can hold what? What would you find in the lacunas? What would you find in them? You would actually find osteocytes in them. Okay. So you would find osteocytes in the lacuna. Okay. Then the lacunas, these little houses, have little networks that connect them to each other, kind of all around. And in the end, these networks, these little channels that connect them to each other, look almost like a Twinkie, the cream rings in a Twinkie. Okay. So these concentric rings, okay, are formed by these connections you can think of. So these connections right here allow for nutrients and waste to be transported, and they allow for the dendrites of the osteocytes to kind of communicate with each other. What are those channels called? It sounds like a Italian dessert. They're actually not the perforating canal. So the perforating canal connects to the central canal and looks like this. So the perforating canal looks more like this. It connects to the central canal and allows the central canal to move outwards. Okay, so that would be the perforating canal. So I'll just do C and put perforating canal. Okay. But back to these channels. These little, yes, canaliculi. Okay. So what letter? We'll do D. Oh, I changed the color. I was on orange. No. D. Okay. So those are the canaliculi. They're networks or channels that connect the lacunas together and allow for transport as well as communication. So D, the canaliculi, and these are network or channels between the lacunas, lacunae, whatever way you want to pronounce it. Okay. All right. Then we already talked about the perforating canal. Now, because of the canaliculi, they form kind of like a ring, a concentric ring. Making it look like the osteon looks like a cut of a tree. So the concentric rings that form due to the matrix, which is the collagen and the calcium phosphate, that's what forms E. And what's E? E are concentric rings of what? Perfect. Okay, so those are the lamella. So let's put E under lamella. Let's fill out the lamella here. And the lamella are concentric rings of matrix. And that should ring a bell, because matrix then is the calcium phosphate plus collagen. Because remember, matrix is made up of ground substance and fibers. Ground substance is the calcium phosphate, fibers is the collagen. Okay, so that's the skeleton, right? Then make sure you go through cells. There are types of cells. We start off with osteogenic cells. Now, osteogenic cells are stem cells. In other words, they're highly mitotic. Highly mitotic. And that means they divide. And when they divide, they're going to differentiate. This is also a common term that we've already talked about, differentiate. And differentiate means it just picks up a specific function, okay? So the stem cells, when they divide, they're going to pick up a specific function and become an osteoblast. An osteoblast, the B in blast is the B in building. These are the bone building cells until they become trapped around the lamella. In other words, they get trapped around the concentric rings of matrix. When they are trapped in one place and they can no longer build, they then differentiate and become osteocytes. Okay, so osteogenic cells divide and differentiate to become osteoblast. Osteoblast builds the bone matrix around itself and forms that lacuna. But once it becomes trapped, it is now referred to as an osteocyte because it's no longer building bone tissue. Okay. Now, osteoclasts are of its own. It comes from a different type of cell, but osteoclast, you want to make sure you pay attention to this right here. This is called the ruffled border, and the ruffled border contains enzymes that break things down. And that's why osteoclast is a bone reabsorbing cell. It breaks the bone tissue down and reabsorbs it. Okay, so osteoclast contains the ruffled borders that contains lysozyme that break things down. Okay, so those are the four main types of bone cells. Then you want to make sure you also know about bone tissue. There are two types. There's compact and spongy. What I recommend you doing is doing a compare and contrast between these two. I'm going through these last few slides a little faster because I want to make sure I have time to go through the muscles, okay? And then under bone formation, there are two types. There's endochondral ossification and intermembranous ossification. So with the bone formation, you don't need to know step by step what happens. You do need to know, what is the starting material? What material does it use in each of these? And then what type of bones result from these bone formation? So for example, if I were to say, okay, long bones are formed by which process? What would you say? Long bones are formed by which process? What would you say? Endochondral or intermembranous? Very good. If I were to say, which of the following uses cartilage in the process of making bone? What would you say? Endochondral or intermembranous? Which uses cartilage? I'll give you, nope, I'll give you clue. It's within its name. Chondrial, the term chondrial, like chondrocytes, means cartilage. So endochondral ossification uses cartilage as its starting material, where intramembranous, it uses a membrane called mesenchyme to build bone tissue. Okay, all right. So what I've done is that I've shared, this is what I've done in my in-class version, kind of like the whiteboard. So some of you might find it helpful. Please use it as you'd wish. We pretty much went through the same thing, but this is just a little bit more of a drawing of it. Okay. For joints, I'm not going to spend too much time on this because it's mostly memorization. The best way to do this is really, and I can't stress this highly enough, is to make cognitive maps. Because you know why? All the terms sound the same. Syndesmosis, synarthrosis, synchondrosis, and so on. So many sins, right? So that's why it's really important to kind of categorize what word goes with what category and also what type. So you can see that synarthrosis is a term that you use to define movement, where syndesmosis is an actual type of a fibrous joint. Okay, so you need to be able to distinguish that. When should I use this term? Because they all sound very similar. Okay, so I filled out the first one for you. I'd like you to fill out the others. Okay, and that way you can just like have a picture, an image of what term goes with what category. And that should help you. Okay. Another important concept in joints is to know the synovial joints. Some terms that you're responsible for knowing within the synovial joints include meniscus, bursa, synovial capsule versus synovial cavity. There's a difference. And then articular cartilage. Okay, so I would go through this, see where they're labeled, and then next to their names, write down their function so that you can really distinguish what is what. Okay, so for the last 15 minutes, I'd like to talk about muscles. So in muscles, make sure you know the function and structure, the neuromuscular junction, the sliding filament theory, and metabolism. A lot of you did your video on these. Sorry, not video, the worksheets you did on these. But I also create supplemental videos on both of these. Okay, so make sure you check out the supplemental videos that I provided on these. And in those videos, I'm pretty detailed about what things are. So just make sure you check out the supplemental videos, and you can find the supplemental videos under announcements. Okay, so go under announcements and then go to either the chapter, the week on muscles, or go through the preparing for your second exam. The supplementals are in both. Okay. So I'm going to spend some time. The request was to kind of go through the sliding filament theory. So I'll go through that with all of you in the last few minutes. But before we do that, before the sliding filament makes any sense, you need to know the structure of a muscle. Okay, so you can see that muscles start out with the muscle group itself. The muscle group is surrounded by the epimysium. And then if I were to tear the muscle apart, it's actually made up of these smaller units called fascicles. Okay, and the fascicles are surrounded by the perimysium. But then if I tear that apart, I break it down, fascicles are actually made up of smaller bundles called muscle fibers. That's really confusing. I want to spend some time talking about that. Muscle fibers, okay? Because you're like, wait a minute. I just learned that fibers were things like collagen, reticular fiber, collagen fiber, elastic fiber, and so on, right? But here I have an actual cell that's called a fiber. And this is the reason why. You can see that that muscle cell right there is long and thin, so it looks like a fiber. So whoever it was did us wrong, but they referred to a muscle cell as a muscle fiber. So when you hear the term muscle fiber, it actually is a muscle cell. So then you're like, wait a minute, then what are the fibers called? Well, the fibers go by another name as well. They're called myofibrils. Myo meaning muscles, fibrils, fibers. So myofibrils are then the actual fibers of a muscle cell or a muscle fiber. Okay? So that terminology just, I don't know who did such a bad decision, but they did. And that's why it can be a little confusing. So whenever you hear muscle fiber, it's actually being referred to as a muscle cell. Okay, it actually is a muscle cell, not being referred to. Where the actual fibers, like the collagen, the elastin, the reticular fibers are actually referred to as myofibrils. Okay, questions? I hear someone unmuted, but I can't hear what you're saying. No? No questions? Okay. Make sure you spend some time on this image because you really need to know what the structure of a muscle cell looks like, or more commonly muscle fiber. So you can see that it has a sarcolemma, it has a sarcoplasm, a cytoplasm, and all of those structures. So make sure you know kind of the structures of what a muscle fiber looks like, as well as the myofibrils. And that's why I had you to do that worksheet. I know that worksheet could be, very difficult, but if it was difficult for you, I would recommend you doing that worksheet a few different times, okay? Until you kind of really, you're able to kind of place what is what and how it fits together, how it works together, okay? All right, so let's go on to muscle contraction, okay? So here's what muscle contraction looks like. You can see that it's divided into 4 main steps. There's excitation. Okay, I don't know why my pen stopped working. It's very inconvenient. There we go. So there's excitation. I'll put my pen. There's excitation, contraction. Then there's contraction, followed by relaxation, which is not on this image, okay? So there's 4 main steps in muscle contraction, or the sliding filament theory. There's excitation, contraction, and then relaxation, which is not in here, okay? So you can see under excitation, the first thing that has to happen is that the central nervous system sends a signal or an impulse to the motor neuron, okay? Now, the motor neuron receives it at the dendrites, passes it through the soma, and then down the axon. When it moves down the axon, it's going to reach the terminal arborization, these branches right here, and then at each terminal arborization, there's a synaptic knob. Okay, so if we zoomed in on the synaptic knob, oops, let me zoom in. So I zoomed in on the synaptic knob. You can see that the signal reaches the synaptic knob, and that causes a chemical to be released. But before that chemical can be released, calcium has to enter into the synaptic knob to cause the acetylcholine to exit. When the acetylcholine exits, it binds the receptors, the green little Y-shaped structure here, to the receptors at the sarcolemma. And the sarcolemma is just pretty much the plasma membrane of a muscle fiber, okay? When the acetylcholine binds the receptors at the sarcolemma, that triggers an electric response along the sarcolemma, okay, which you can see right here. So the signal then travels down the sarcolemma until it reaches the transverse tubules, or the T tubules, that allow it to get deep into the muscle fiber. When it gets deep into the muscle fiber, it's able to then stimulate this structure right here. This organelle right here is called the sarcoplasmic reticulum. Essentially, that's the smooth endoplasmic reticulum. And same, just like when we studied in organelles, the smooth endoplasmic reticulum contains calcium. So when that signal
Oh
reaches,
no, I'm taking a minute 4 o'clock today, but
so when the signal reaches the sarcoplasmic reticulum, it causes the calcium to be released into the muscle fiber. When the calcium is released into the muscle fiber, the calcium binds onto the troponin and tropomyosin structure, which I think I drew down here. Here we go. Okay. So when it binds to the troponin and tropomyosin structure, that allows the troponin and tropomyosin to shift away from actin. And when it shifts away from actin, myosin heads can grab onto the actin and pull, and that's when you have contraction. So we know that a muscle starts off as a muscle group right here. Right. So the muscle group right here. And the muscle group is surrounded by, so the muscle group is drawn like the piece of thigh right there, you know, and then surrounding the muscle group in the green is the fascia. The fascia is the connective tissue that surrounds it, or the fat. And the fascia that surrounds the muscle specifically is called the epimysium. Okay. Now, if I were to break the muscle down into its smaller units, then I'd have what's called fascicles. So all of these red bundles right here, these little red circles, those are fascicles. Okay, now fascicles are surrounded by the paramysium, peri meaning around, mysium muscle, so it surrounds the fascicle. So the green right there, the green right here is the paramysium, where this right here is the fascicle. Now you can see that within the fascicle, there's even smaller bundles. If I pulled one bundle out, then I'd have a muscle fiber. And like I said earlier this morning, technically it's not a fiber, because remember, fibers are things like collagen, elastin, reticular. But because this structure looks so much like a fiber, it's called a muscle fiber, although in actuality it is a muscle cell. So this is a muscle cell that's called a muscle fiber because it looks long like a fiber. Okay, so then you're like, well, what is a muscle fiber then? Well, they go by a different name. They're called myofibrils. And the myofibrils include things like actin, myosin, and so on. Okay, so you have the muscle fiber, which technically is the muscle cell. It is also surrounded by a fascia, a lipid called the endomysium, endo meaning the innermost. Okay, so muscle groups or muscles are surrounded by the epimysium. Fascicles are surrounded by the paramysium, and a muscle fiber is surrounded by an endomysium. Now, if I zoomed in on the muscle fiber, which is the cell, it contains all the organelles that you learned about in chapter 2. So it has the plasma membrane, except that someone tried to be tricky and wanted people to learn a different name. So the plasma membrane of a cell, okay, if it is a muscle fiber or a muscle cell, it goes by the term sarcolemma. Okay, so we don't use plasma membrane anymore. Even though it is a plasma membrane, we call it the sarcolemma. So the sarcolemma is the plasma membrane of a muscle fiber. Okay, so give yourself some time to kind of wrap around the terminology that we use. Okay, so the sarcolemma is the plasma membrane of a muscle fiber, where in a normal cell we call cytoplasm. Here we call it sarcoplasm. So the sarcoplasm is the cytoplasm, everything inside of a muscle fiber. Okay, so inside of a muscle fiber, aka cell, you would find similar organelles. Okay, you would find this organelle right here. What is this organelle right here? What is that organelle? I'll give you a clue. In chapter 2, you learned about this organelle, and you learned that it stores minerals, it synthesizes lipids, and it detoxifies. What organelle am I talking about in a normal cell? Yeah, exactly. So in a normal cell, this would be the smooth endoplasmic reticulum. But just to make us learn a little bit more, somebody one changed its name and now it's referred to as the sarcoplasmic reticulum. And that's why oftentimes you'll see me abbreviate it as the SR. But what is the SR? Essentially, it's the smooth endoplasmic reticulum. Functions the same way. It stores minerals, and specifically, it's going to store calcium. It's going to be able to synthesize lipids, and it's going to be able to detoxify. Okay, so that's the sarcoplasmic reticulum. Then you have to have energy, right? You have to have energy, and what organelle produces the energy? So in a muscle fiber, aka a muscle cell, you will find lots of these organelles. What organelle am I pointing to? So it produces ATP. There you go. Thank you. This is called the mitochondria. So you will find lots of mitochondria in a muscle fiber. Okay. Now, in order for the mitochondria to produce energy or ATP, like one of you had said, you need two things. What two things do you need to make ATP? What two things do you need to make ATP? This one was in muscle metabolism, if you can recall from your studying. In muscle metabolism, yes, very good. You need glucose, okay? Now, in a muscle fiber, glucose is stored in a chain of glucose. What is a chain of glucose called? So each of these little black dots right here is one glucose. But if you have a chain of glucose, what is that called? Glycogen, exactly. So all of those little dots that form a chain, that is representing the glycogen. So a muscle fiber would have lots of a muscle fiber would have lots of glycogen. What's the other important ingredient that you need to produce high amounts of ATP besides glucose? Yes, very good. Now, oxygen is going to be stored on what protein in a muscle cell or a muscle fiber? So these red dots right here represent this protein that can store oxygen. What are these proteins called? I'll give you a clue. These proteins in blood is called hemoglobin. Yes. So in muscle, it's called myo meaning muscle, globin meaning protein, right? So here it's going to be called myoglobin. Okay. So those are the basic organelles or structures that you would find in a muscle fiber. You would find lots of mitochondria. You'll find lots of sarcoplasmic reticulum, lots of chains of glucose in the form of glycogen. You'll find lots of myoglobin. Okay. Now, the sarcolemma has two distinct areas. Okay. One distinct area on a sarcolemma is this area right here and this area right here. Okay, so the sarcolemma has two very distinctive areas. One area is really important. Why? What are those green little Ys represent? What are those green little Ys represent? Receptors, exactly. What are they receptors for? Yes. Okay, so they're receptors for acetylcholine or ACH. Okay, now this area, your textbook calls it the junctional folds. But if you went to school about 20 plus years ago, like I did, we used to call it the motor end plate. Okay, so that area where you find receptors for acetylcholine in your textbook is called the junctional folds, but it used to be called the motor end plate. Okay, so that's one specialized area of the sarcolemma. The other specialized area of the sarcolemma is where it dips really deep down and it forms a T. What is this area referred to as? The transverse tubules. Very good. Or the T tubules. This morning, I kind of went through the steps and I'm like, I'm not sure that they know where these things are because I didn't talk about them yet. So that's why this time I'm going to talk about all the parts so that when I go through the steps, it should make a little bit more sense. Okay. So what we just talked about right now is just one layer. Okay. That's just one layer of the muscle fiber. Now I'm going to add another layer to the muscle fiber. The next layer that I'm going to look at involves the myofibrils. Okay, same muscle cell or muscle fiber, but now we're going to add a layer of the actual myofibrils. Okay, so muscle fibers have several proteins or have several myofibrils. Two main myofibrils are the thin and thick filaments. So in here, you see these lines right here. There's one there, there's one there, there's one here, there's one here. Okay. So what you can observe about these are that they are thin and they're always connected to the zigzag structure right here. This zigzag structure is called the Z bands or the Z lines. Okay. So what filament is thin and attached to the Z lines? What myofibrils is that? Actin, very good. So what I did is that I zoomed in on one actin, and this is what the actin looks like over here. So I zoomed in on one actin, and this is what the actin looks like right here. So you can see that on actin, there are these little white holes right there that's currently covered by this green and by the red. Okay, so I zoomed in on the actin, and actin actually has these areas where we call them myosin binding sites, but those myosin binding sites by default automatically are covered by the troponin and tropomyosin. Okay, technically they're covered by the tropomyosin, and then attached to the tropomyosin you'll find troponin. Okay, so together we call it the T&T system. Oops, didn't mean to use the highlighter. Let's use this. So together we call it the T&T system. Can someone tell me what the function or what's the purpose of the troponin tropomyosin system? What does the troponin tropomyosin system do? What's their function? They can allow for calcium binding. Yes, definitely. So calcium does bind to the troponin. Yep. But what's their function though? What are they supposed to do? What's their purpose? Yes, they regulate the interaction between actin and myosin, and specifically, Sean's right, they prevent contraction. Okay, and think about it. If you're sitting there right now, you're wherever you are, your muscles are by default relaxed, right? You're not all crunched up and contracted all the time, right? That would take too much energy. So by default, until your brain tells a muscle to contract, by default, your muscles should be relaxed. And they're relaxed because the troponin tropomyosin system prevents contraction. That's their purpose. The troponin tropomyosin system prevents contraction. Okay, because just think, you don't want your muscles to be contracted every single minute of the day, right? You'd expire from using too much energy, you'd be too tired, and you'd have muscle fatigue. Okay, so the function of the troponin tropomyosin is to prevent contraction. Okay, so then when you do want it to contract, that's when you have to get tropomyosin to shift away from the binding sites, right? And to get that to happen, troponin has to pull on tropomyosin, and troponin will only do that if what is released to bind to troponin. Yes. So you need calcium to allow for contraction. Okay. And let's walk backwards. Where would that calcium come from? What did we just cover that carries calcium? What would have to release? Yes, the sarcoplasmic reticulum. Exactly. Now, normally the sarcoplasmic reticulum doesn't open. It doesn't allow calcium to flood into the sarcoplasm. So what has to happen in order for the sarcoplasmic reticulum gates to open and allow calcium to flood the sarcoplasm? A signal, right? A signal. And where's that signal going to come from? An action potential. Excellent. Okay. So sometimes it helps to actually walk backwards to understand the steps more than to actually go through step by step in sequential order. Okay. So when you do it this way, you don't have to memorize it. It's logical. It makes sense that this has to happen before this has to happen before this. Okay. So I don't want you to have to memorize the steps in muscle contraction. It should just make sense if you walk yourself backwards on how muscles can contract. Okay. All right. Okay. So an analogy that I often give is contraction is like Romeo and Juliet hooking up. Okay. So Romeo and Juliet is actin. Okay. And Juliet has very protective parents called the troponin tropomyosin. And Romeo, which we'll call myosin, wants to hook up with Juliet, which is the actin. But her parents won't let her hook up. Okay. But it turns out that her parents love chocolate. If you give them chocolate, they're so busy eating that chocolate that they're not really watching over Juliet. Okay. And that's why you have to give the chocolate, aka calcium, to distract the troponin tropomyosin so that Romeo can hook up with Juliet. Okay. If that works for you, you go with it. Or if the straight science works for you, whatever works for you. But that's the things that I do to kind of help me remember. I make a lot of mnemonics, little stories to kind of help me remember things. Okay. So now that we kind of get the gist of things, I want to talk about now the actual contraction. Okay. All right. So now when I go through the steps, hopefully it makes a little bit more sense than me just reading off here. Okay. So to start everything, because remember, your muscles aren't by default contracted. Your brain has to tell your muscles to contract. And that's the first step. So in the first step right here, the central nervous system, the brain, has to send the signal through an action potential through a motor neuron. Why a motor neuron? Why not a sensory neuron? Why not an interneuron? Why a motor neuron? Who can tell me that? Why through a motor neuron? Why an action potential through a motor neuron? Because, yes, exactly, because of the direction the neurons are located, right? Motor neurons always carry information from the central nervous system and sends it out to the effector. And the effector in this case is a muscle, where sensory neurons detect things from the environment and sends it to the central nervous system. Because information only travels in one direction along a neuron. Motor neurons carry direction out, sensory neurons carry it in. And that's why the central nervous system, in order to send out the signal, it's got to go through a motor neuron, okay? So it sends a signal along the motor neuron. It reaches the terminal arborization, where you have the splitting. And then let's say it goes to this one right here. It reaches a synaptic knob. Right here, okay? So when it reaches the synaptic knob, let me zoom in. When it reaches the synaptic knob, okay, now we're at the point of the neuromuscular junction. Okay, so we at the point of the neuromuscular junction, the place where the neuron meets the muscle. Okay, so the neuromuscular junction is the location where the neuron meets the muscle. Now, notice the word J in there. That's the junction. That implies that there's a space. Okay, now, a neuron never actually comes into direct contact with a muscle. Okay, so a neuron never actually comes into direct contact with a muscle. Now, whatever device you're on, hopefully you're on a device, hold the wire for me. Okay, if you can hold the wire right there, okay? If it didn't have that plastic around that wire and you touched it, what would happen? What would happen? Okay, so here's my wire right here. Oh, you can't see my wire. My camera's all the way over here. Okay, let me grab my camera. Okay. There you go. So here's the wire, right? Exactly. So if it wasn't the plastic here, if you touch it, then you would get shocked. But if I go like, so here's my wire. There we go. If I don't touch it, if my finger is like right here, would I get shocked? No. You have to have direct contact for electrical impulses to pass. Do you see that concept? For electrical impulses to move from one structure to the next, you have to have direct contact. Okay? But what I'm saying is that the neuron never comes into contact with the muscle. So how the heck is the signal going to cross? How is the electrical signal going to cross? Because electricity requires direct contact. Exactly. That's why I had you learn about the chemicals, the neurotransmitter. Because there's no direct contact, which is required for electrical, we have to send a chemical signal. And that's why at the end, right here, the synaptic knob, once the signal is sent, the synaptic knob is when the signal reaches the synaptic knob, calcium diffuses in. I'm going to turn off my camera now. Okay, so calcium diffuses in, which is what you're seeing here. Calcium, oh gosh, honey, what happened? Here we go. Calcium is going to diffuse in, and when calcium diffuses in, acetylcholine is pushed out. Okay, so calcium is going to diffuse into the synaptic knob, and acetylcholine is going to be forced out through exocytosis. When the acetylcholine moves out, it's a chemical. It can travel, right? It's going to travel across the synaptic cleft or the junction, and then that's when it binds to these structures right here, aka the receptors along the sarcolemma. And that's how the electrical signal is able to jump the junction, the space, and go back to being electrical signals again. And that's why you had to learn about how it went from electrical to chemical back to being electrical, because the neuron never comes into direct contact with the muscle. Okay? That's why it's important to understand the role of acetylcholine, the neurotransmitter. Okay? So the signal regenerates along the sarcolemma. Then it's going to move down the T tubules. Why is it important to move down the T tubules? Why is it important to move down the T tubules? Yes, exactly. Because the sarcoplasmic reticulum is deep down. You can see in this image right here. So the signals move down the T tubule so that it can reach the sarcoplasmic reticulum. Why is it important to reach the sarcoplasmic reticulum? You know this now. Why is it important to reach the sarcoplasmic reticulum? What does the sarcoplasmic reticulum have that you need for contraction? That's right, calcium. Okay? So here, again, calcium plays a role. Okay? So at this point, the calcium diffuses into the muscle fiber. And what's the purpose of that? Why do you want calcium into the sarcoplasm? Remember the story I told you earlier? Why do you need calcium in the sarcoplasm? Exactly. To hang with Juliet, to distract Juliet's parents. That's exactly it, to distract Juliet's parents. So the calcium binds the troponin, troponin pulls tropomyosin, and now guess what's exposed? The ladder, right? Now actin binding sites are open to myosin heads. Okay, so now the myosin heads are going to bind to actin, and that process is called cross bridge formation. I want you to know that term, cross bridge formation. So cross bridge formation just means that the myosin heads are now able to grab onto actin. Okay, and that's when you have a power stroke occur. In other words, myosin heads pull actin inward. But wait, before heads can pull actin inward, it's got to have energy, right? It's got to have energy. So the myosin heads have to consume a molecule of ATP before it can cock and bind to the actin, and then it pulls. Every time it pulls, it needs one molecule of ATP. So you can imagine how much energy is needed for contraction to happen. Okay. Each time it pulls, it gets closer and closer and closer until it completely contracts. And then when it completely contracts, you have the shortening of the sarcomere. And the sarcomere is the space between two Z lines, and that is contraction. All right. Now, you don't want to stay contracted. Eventually, you're going to want to relax. Okay. When you're ready to relax, the brain is going to stop sending the signals to contract. There's no more signals moving down. Let's use pink. There's no more signals moving down the neuron. No more signals moving down the neuron. No more calcium entering. No more calcium entering. No more acetylcholine being released. Oh, good. Okay, so we've stopped contraction, technically. But what happens to acetylcholine that's currently there? You have to have something to remove the acetylcholine from the receptors that is currently there. Exactly. You need an enzyme called acetylcholine esterase. So acetylcholine esterase, ACHE, breaks the acetylcholine down so it's no longer bind to the receptors. When there's no longer acetylcholine binding to the receptors, no more signal is being generated along the sarcolemma. No more signals, no more moving down the T tubules, no more causing the sarcoplasmic reticulum to release calcium. Yes, we're almost there. No more release of calcium, but what's the problem? So no more new calcium being released, but what's the problem? I still have the calcium that has already been released, right? So in order for the final steps of relaxation to occur, I have to actively pump that calcium back into the sarcoplasmic reticulum. Okay, so I have to actively pump the calcium back into the sarcoplasmic reticulum. And now, guess what happens? No more chocolate. Parents aren't distracted. So now they're like, They go back to covering the binding sites on actin. Juliet is like, oh, sorry, dude. And Romeo is like, oh, can't hang on to her anymore. So then it can't bind. So then you have relaxation. Okay. So let me ask you a few cognitive questions. Okay. How many times did you have to use calcium in this process? The entire sliding filament theory? Twice, exactly. Can you tell me when and when? Exactly. Yep. So the synaptic knob, in order for it to release ACH, calcium was a role there. And then you had to use calcium again in order for contraction to happen because it has to bind to the troponin. Great. How many times did you have to use ATP? I understood you. It's all good. How many times did you have to use ATP for the whole process? Three times, exactly. So tell me when. When did you have to use ATP? Anyone can answer. Don't forget, we have to talk about the nervous system too, so don't wait too long. Breaking myosin and actin. Binding, yes. So you need ATP to activate the myosin head so that they can bind to actin. Yep. I understood you as well. So good. Two more. Opening the gates. What gates? Exocytosis of ACH. Excellent. Exocytosis is an active mechanism that requires ATP. Absolutely. What do you mean by opening gates? I think you're close. Yes, exactly, Caitlin. You need energy to shuttle the calcium that's in the cytoplasm or in the sarcoplasm back into the sarcoplasmic reticulum. That's exactly it. That's the 3 times in which you need ATP in this whole entire process. You understood. Great job. Okay. All right. I think you got it. You have a great understanding of it. Okay. So I'm going to move on to the nervous system, if that's okay. We only have 20 minutes left. So I want to make sure I start, I have enough time on the nervous system. Okay, so for the nervous system, I feel, oh, that's okay. So for the nervous system, one of the things you'll need to know is the division of the nervous system. So I took this from a whiteboard drawing I made for my in-person class. So you can see that the nervous system is divided into the central nervous system and the peripheral nervous system. The central nervous system includes the brain and the spinal cord, where the peripheral nervous system is first divided into sensory and motor. We talked a little bit about that, right? You have the sensory neuron, you have a motor neuron. Now, within the sensory neuron, you have what's called the somatic and the visceral. Somatic oftentimes is associated with voluntary, and visceral is oftentimes associated with involuntary, okay? Then the motor is also divided into the visceral and somatic. And if it's a motor neuron that has somatic control, oftentimes that means it's going to a skeletal muscle because skeletal muscle is voluntary control. Where if it's a motor neuron going to your organs, internal organs, we call that visceral, and oftentimes we do not have voluntary control over that. We call it the autonomic nervous system. So the visceral motor neuron is often associated with the autonomic nervous system because it is overall involuntary. Now, the visceral motor nervous system is divided further into the sympathetic and the parasympathetic, both involuntary, but one is referred to kind of your fight and flight response. In other words, you're kind of on hyperactive attention, okay? Where the other one is called your parasympathetic or your rest and digest, where you sense, there's not much danger, so you can pool all of your energy into kind of resting and digesting food, okay? So that's the major divisions of the nervous system. You also want to know the spinal cord as well as the neuron itself and the brain, okay? And you did that in your activity, right? Your assignment. So you had to go through the parts of the brain, which include the cerebrum, the diencephalon, the brainstem, and the cerebrum. And then I asked you for subregions. So subregions of the cerebrum would have been the lobes, the frontal, parietal, temporal, occipital. The subregions of the diencephalon include the thalamus and the hypothalamus. The subregion of the brainstem include the pons, the medulla oblongata, and the midbrain. Okay, and the cerebellum is just the cerebellum. We didn't really go into any more subdivisions of that. So make sure you know the parts and the functions and all their specific information. For the spinal cord, make sure you know the parts of the spinal cord, be able to tell what's the posterior versus what's the anterior. You can tell that because the H, you can see that the H, let me draw that out. So here's this butterfly shape right here. This butterfly shape right here. That's the gray matter. And you can find that the butterfly shape is thicker on this portion right here and this portion right here. And that's always going to be the anterior. Okay? Where the thinner portion of the gray matter is always the posterior. That should help you kind of remember, okay? Make sure you know the spinal cord and nerves. There's 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccyx. Know parts of the spinal cord, for example, the anterior root, the anterior root, and then the posterior root ganglion, the gray matter, the white matter, the central canal, and so on, okay? And then know parts of the neuron, the dendrites, the soma, the axon. Now the axon does have additional parts, right? So the axon has the axon hillock, it has the length of the axon, and then it has the terminal arborization, and then you have the synaptic knob. So make sure you know the parts and the function of a neuron. Okay. Here's where students usually ask me to spend some time on, and that's the nerve impulse conduction. Okay, so in the nerve impulse conduction, there are four main events that you need to know. One, resting membrane potential. Two, local membrane potential. Three, threshold. And four, action membrane potential or action potential. Okay. In resting membrane potential, we refer to that as polarized. What does polarized mean? Well, sorry, I have it written right here. It means that it is negatively charged. Okay. Polarized means it is negatively charged. So if I drew a neuron, okay, inside, what ion do you have on the inside of a neuron? Yeah, potassium. Potassium, is that positive or negative? Is positive, exactly. Wait a minute. I just said at resting membrane potential, it is negatively charged. That's because even though potassium is the major ion that you find in a neuron, you actually find a lot of negative proteins. Okay, and these negative proteins are large and strongly negative. These do not move. They're always in the neuron. Okay, and that's why even though there's potassium inside the neuron, there's just more strongly negative charged proteins. And that's why overall, if I were to measure a neuron at resting membrane potential, it would read negative what? And you should know these numbers. Negative what? Negative 70 millivolts. Very good. Okay. So at resting membrane potential, it's polarized because it's at negative 70 millivolts. Why is it negative 70 millivolts? Well, it has potassium, but it also has strongly negative anions. Here are proteins. Okay. So at a local membrane potential is when it receives a stimulus. It receives a stimulus. Okay. But what are some of the characteristics of a local membrane potential? Local membrane potentials are not action potentials. Okay. Local membrane potentials are different in the following. Yes, very good. I like that word. It's graded. Okay, meaning that it has different levels. Okay, what else? It's decremental, very good. And what decremental means is that as it gets further away from the beginning, it starts to lose. It starts, no, I wouldn't say lose, it starts to weaken. As it gets further away from the area of stimulus. What else? One more term. Yep, it cannot go long distance. Yep, that's part of the decremental. Very good. What else? Yes, it's reversible. Okay, so a local membrane potential. Oh, why is my highlighter not working? There we go. A local membrane potential only occurs right here. Okay, it only occurs along the dendrites and the soma. It never reaches the axon unless it reaches threshold. Okay, so local membrane potential is graded, decremental, and reversible. And it never reaches the axon unless it achieves threshold potential. Okay, threshold potential means that it's strong enough to be sent along the neuron. In other words, it's strong enough to reach the axon hillock, the beginning of the axon. Okay, so the axon hillock is right here. The beginning of the axon. In order to reach the axon hillock, it's got to change that membrane potential from -70 to what? It's got to be strong enough to -55, essentially, is exactly. So it's got to go from -70 millivolts to -55 millivolts. In other words, it's got to have a 15 millivolt charge. Okay. Once it reaches that -55 millivolts, then that signal is converted into an action potential. Okay. And how an action potential is different from a local membrane potential, instead of being graded, decremental, or reversible, it is an all or nothing. Think like once you reach the top of a roller coaster, once it goes, there is no stopping. You can pee your pants, you can scream until you have no voice left. Once it goes, it goes. That signal is going to be sent. It doesn't matter how weak, how strong, it's going to go. Okay, so those are some characteristics, the different characteristics between a local membrane potential and an action potential. Two main differences, right? One, the characteristics are different. Two, the location is different. A local membrane potential only occurs along the dendrites and the soma, where an action potential only occurs along the axon. And it's once it goes, it's going to go. It doesn't get weaker. It doesn't go backwards. It just continues. Okay. So in order to understand that, we have to understand what does it mean to be depolarized and repolarized. Okay. So imagine this. You have a ball, a plastic ball on top of water. If that water is stagnant, does that ball ever move? Imagine a plastic ball on top of stagnant water. If that water does not move, does that ball move? No, it doesn't. Okay, so here's some water, and here's my ball, right? So as long as there's no waves in that ball, nothing happens. The ball doesn't move. If I want that ball to move down to the next place, I have to have an up wave and a down wave. Up wave, down waves. I have to have a change in membrane potential in order to move that ball from one location to the next, to the next, to the next. And that's why I have depolarization, repolarization, depolarization, repolarization, depolarization, repolarization. So in an action membrane potential, one area undergoes depolarization and repolarization to move the ball along. What's depolarization is when the membrane becomes what? So if polarization is negative, D is the opposite. So depolarization means the membrane is becoming what? Positively charged. Exactly. How does it become positively charged? So I've got my potassium. I've got my negative anions or negative proteins. How does it become positively charged? Yes. In depolarization, sodium enters into the membrane, causing it to become positive. How do I cause it to become repolarized? Polarized meaning negative, re meaning returning. What does it mean to be repolarized? Yep, the potassium exits, but what in terms of charge is happening? Yes, it's going back to being negative. How does it become negative? Sean said that, right? So now we have the potassium gates open and potassium leaves. It's like, what? I was positive so short and now I'm negative again. Okay. And here's the problem. It's not a problem, but this is what happens. Okay. For every 2 sodium that enters, you have 3 potassium leaving. So that's why not only does it get negative, but it gets even more negative than normal. We call that hyperpolarization. Okay. So it goes from depolarized to repolarized and actually to hyperpolarization. Just because more potassium, in the exchange, you have less sodium coming in than potassium leaving. Okay. So that's what happens, but it happens from this area of the membrane to this area, to this area, to this area, until finally the signal reaches what? It's already moving along the axon, but it's going to reach what? Nope, can't be the dendrites because the dendrites are over here. There you go, the synaptic knob, exactly. And guess what? That connects to our story of Romeo and Juliet, right? That's the neuromuscular junction. The neuromuscular junction is an example of what continues next after all of this happens. Okay, so when the signals reach the end, calcium enters into the synaptic knob, neurotransmitters such as acetylcholine exit, and then acetylcholine binds the receptors along the sarcolemma, blah, 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 so on. And you know the ending of that story. Okay, so that's nerve impulse conduction. All right, here's another diagram of that. You can see a resting neuron is positive on the outside, negative in the inside, even though you do have potassium on the inside. Then you have an action potential. It totally skipped a local potential, but anyways. So went to an action potential. In an action potential, you can see that one area is becoming depolarized, repolarized. Then the next area is becoming depolarized, repolarized, and so on until the signal finally reaches the synaptic knob. Okay. Okay, and that's what I drew over here. Okay, so what we just talked about is the electrical physiology. We talked about resting membrane potential. We talked about local membrane potential. We talked about threshold, and we talked about action potential. Okay, and here's what it would look like if you were to look at a graph of it.